예, 그라니에 백
Dr. GRANIER is in charge of the coordinating the scientific expertise at ANSUS, and she has been focusing on AMR and other research projects at ANSUS. And she is also the charge of FAO AMR Reference Center for France. And let's welcome her with the topic of application of WGS for AMR surveillance along the food chain. Please welcome her with a big round of applause.
Good morning, everyone. I would like first to thank the organizer for giving me the opportunity to join you today and talk with you. The question that I was given when we first get in touch was, can you do something about application of whole genome sequencing for AMR surveillance? First, I thought, would I give a global presentation, but there are people much better than me to do so. A European perspective, and I think it would be a bit too much. So I'm just going to talk about what I know best, surveillance in my country, France. First of all, I'll start with a disclaimer. What I'm going to present you is only my views. It is not my institute, it is not my government opinion, it's just my opinion that I want to share with you. And before we share my opinion, and we don't know each other, I think I have to introduce myself. So my university background is molecular biology, but I have a PhD in microbiology, and I spend most of my time doing traditional Pasteurian microbiology. I worked a lot I've been very much involved in the EU harmonized AMR surveillance program along the food chain that is regularly published and a gold mine of information. And very recently, I've been asked to join the team of coordination of FAO Reference Center for France. And that's it. Now, I said that it's not the opinion of my agency, but give me a few minutes to present you in 3 slides what ANSES is doing. So ANSES is a French scientific expert appraisal agency. We work in the field of our slogan that we use all the time is investigate, evaluate, and protect. We are supposed to help the decision makers with scientific-based information, and there are so many different expertise in our institute. We work on human health, animal health, plant health. The agency was created in 2010. We are 1,400 employees. Our goal is to produce knowledge, and we are deployed all over the territory. It is for production purposes. Where the animals are, that's where the labs are. We are doing a lot of reference activities. Last but not least, because it's in line with our subject today, one of our missions is vigilance and alert. So we've got 5 vigilance schemes, and I see that it's very close from what you are doing here. Toxicovigilance, nutri-vigilance, phytovigilance, veterinary pharmacovigilance, and also preventing occupational health diseases. We have also numerous 3 platforms dealing with human health, plant health, and food chain safety that are gathering public and private efforts and makes the information circulate fast. We have surveillance networks. And one we're going to talk about it later is really focused on antimicrobial resistance, and it's dealing with animal diseases, sick animals. It's called RESAPAT. We have a network on Salmonella, which is quite historical, and also another network on Mycoplasma. And we have observatories that collect data on food quality, table of composition. So that's it. Now you know who I am and who I work for. Let's go back to our question. When talking about surveillance, my first idea was to check what we are talking about. And if you bear with me, I chose to pick the WHO definition of surveillance, which is published in this international health regulation, and I think it will help to focus our talk later. So surveillance means a systematic ongoing collection, collation, and analysis of data for public health purposes, and timely dissemination of the public health information for assessment and public health response as necessary. There is plenty of things in this definition, plenty of requirements that would be important for the rest of the talk. May I introduce this paper made by some colleagues. The first author is from ANSES also. In this paper, they try during year 2021 to determine in a World Health perspective all the surveillance program that were running in France at that time. And they identified 48 different surveillance programs dealing either with antimicrobial resistance, antimicrobial use, antibiotic residues. They were gathering information about human, animals, and their environment. So far, so good, really a One Health perspective. But if you get into deep, out of the 48 programs, there are 35 programs that are collecting antimicrobial resistant data, and 31 out of them were collecting data related to clinical human samples. So you see that the balance is not really okay. In this paper, there is this nice overview of the 48 systems that they identified. Let me walk you through this figure. So if you look, let's start with the first column in blue. It's going to be the human data. The second column in yellow, going to be animal and food data. And the green column, the last one, would be environmental data. Then let's walk through the lines. The first line is about antimicrobial resistance. The second line is about antibiotic use. And finally, the third line is about antibiotic residues. You see that most of the network, as I said before, are focused on the left-hand corner of this table. So I've been in detail into this paper to try to find whose network are gathering at least a little data by WGS. And what a surprise, there is only 7 that are marked with the red cross that are collecting WGS data. And okay, for most of them, they are what we call in Europe National Reference Center, means that they are in charge of human health only. That's what you have on the top blue corner. Some of them, like the Salmonella and the Listeria, they are mostly WGS based and gather all information over there. But if you look at the IMR data that are nowhere, that are produced by ANSES, either the National Reference Laboratory IMR on the bottom yellow or the RESAPAT network about sick animals, they gather phenotypic information, and they are doing WGS on a very special occasion on special phenotypes. So we cannot say that the surveillance by WGS is really in place for IMR so far in my country. So, what is the situation in Europe and for the food chain? So I've been digging into those big and fantastic reports by IFSA and ECDC, published on a yearly basis with data from food, animals, and humans. And first of all, my first approach was, let's do a search in these documents. So if you look at the document published in 2022, very recently, gathering data from 2019 and 2020, you can see that I found only 31 hits when I search for WGS or whole genome sequencing. That's not a lot out of a 200 pages report. But 2 years after that, if you look at the report published in January, the situation is much better. At least I got 116 match. Why that? And why it's still not, it's still phenotypic data and not a lot of WGS data. Since 2021, we have started to gather information on a voluntary basis, but it's not all the data. I cannot provide the antimicrobial resistance of the Salmonella for France by WGS for the moment. They are only collecting the data for ESBL, AMC, and carbapenemas producing Salmonella and E. coli, which is just a little part of the data set. But at least we are starting to work together. We are starting to collect WGS good quality data. I'm a molecular biologist, and we are talking about this WGS surveillance program for so many years. So my next question was, why does it take so long? It's unbelievable. And I recommend that you might read this paper published in 2017 by UCAST, which is a gold mine if you are dealing with phenotypic studies on antimicrobial resistance. You find everything and a lot of information. So one of those groups really worked on whole genome sequencing for antimicrobial susceptibility testing. And we have to tell immediately that maybe one of the big problems is that WGS is absolutely fantastic to assess, to study, to follow resistance mechanism. But it's definitely not good to predict susceptibility. But predict susceptibility is what our clinicians want. When they want to prescribe to an animal or a human an antibiotics, the only information as they are interested in is, will the treatment be okay? Will it work? Is it susceptible? But us, doing surveillance, epidemiology, we are, in fact, only are interested in resistance. Follow the mechanism, follow the ESBL through the animal herds, or trace some foodborne outbreak, I don't know. But our markers are resistance. And unfortunately, for many, many years, we have gathered phenotypic study because most of the time they were done for clinical purposes. We are just catching the data afterwards, after it has been used as individual test, and make statistics about that. So I guess we have also to tell to our clinician that it won't help, WGS don't help to, for the moment, to predict susceptibility. Maybe it will do better for the moment. Please prove me I'm wrong, but I'm not convinced at all that it will help to predict susceptibility. And we have to admit that, well, traditional Pasteurian microbiology is considered to be slow. WGS is much more slow. It's not fast. And also, I think one of the limitations is that who's going to pay? Because when we were collecting clinical data, they were paid by the system, they were paid by the farmer. But this surveillance, not linked to any clinical event, should be paid by someone. Also, and I've been into that group, we told to our stakeholders that WGS was the gold standard, that it was easy, fantastic, reliable. No. And since we are using it, we must say that it's not. not really. We need to work on it. And very recently, in September, this paper was published by the team of René Hendriksen. I think it was here last year for the third edition of this conference. So in this article, they did a proficiency test for 2 years in a row. And they have national reference laboratory from different European countries participating. I think it was 16 the first year and 19 the first year. My team was participating to this trial. And we discovered that the results are not that good. There is a little work to standardize and to do, again, some laboratories were really outliers. And they conclude this article that by saying a nice sentence about challenging of dealing with the contamination. Okay. So my image, the image I gave you is quite bad. I want to be optimistic. There are plenty of things published, new information, new progress. On the top left corner, I want to show you that there is an ISO-certified workflow to analyze the data and get to the detection of the mechanism, and it's quite reliable. In the meanwhile, ISO set a group, it was a year ago, to work on AMR testing, determine what's going to be the future, try to be a bit in advance and prioritize. Would it be Malditoff? Would it be WGS? Would it be metagenomics? Would it be? Everything was open to discussion. And the top priority is WGS method. And the top priority inside the WGS method is to certify the way we're going to identify the gene, identify the mechanism. So the project is on its way. Bear with us. I'm happy to be part of this group. And I think it's a long way, but there is a strong will of collaborating and writing something clean to have something harmonized. Go back to René's team paper that I shown on the slide before, the bad point of the study. But you got to be optimistic. Within 1 year, some labs really improve, which means if we work together, if we standardize our method, and if we understand what are the problems, we make progress, so it can be fast. So, standardization of all the steps of the use of WGS, it's on its way. Don't forget, for the moment, please, with WGS, focus on resistance and not on susceptibility. It would be dangerous. Costs are going down. Quality of the data are going up. Our experience in the lab are going up. So there is everything to say now to my question. We are not there yet, but very soon we should be. I want to thank my colleague from ANSES for constant discussion, and again, the organizing committee to have invited me here, and I'm ready for your questions.